Hello there, ninth graders. This is Teacher Krishna, and welcome to our biology class for week number 18. Just like every week, thank you for coming and, of course, for completing all of the assignments that we have prepared for you. So, let's follow the same instructions as every week. Log into Agilex, log into Google Classroom, download your Word document, and save it inside your science or your biology folder, just like it is specified right below. It's very important to start with our ending mind, so today we are studying topic 3.04. And the ending mind is the following. Students will be able to create a compilation of all the summaries of the unit to have resources to study for the final quiz of the unit. So we are going to study one last topic, and then I'm going to give you some instructions about how to create your summaries, of course, or how to compile them, most likely. All right, so today we're going to be analyzing heredity patterns. And for that, we have to analyze two very important words, which are phenotype and genotype. So, before we analyze these two words, we have to answer to this question. How can probability relate to heredity patterns? So, let's imagine that you have a coin with you. In the coin, you have heads and tails. So, what happens if you flip it one time? Well, you are going to get 50% chances of getting heads and 50% chances of getting tails. So something like that is going to happen whenever we recombine different traits and whenever we recombine different alleles as well. So we are going to analyze it a little bit more later on, but you will find out that some traits are going to be more likely to appear since they are dominant than recessive traits. And here we have the first important term, which is homozygous. What is an homozygous organism? It is an organism with two equal alleles. Okay, so if you see the picture that we have right on the left, you see that the first one has P and P. It has both capital P's, which means that it has the same allele. So when they are equal, that is an homozygous organism. Okay? But what about a heterozygous organism? It is an organism with two different alleles. Right, for that we can check the second flower, in which there is a capital P and a lowercase p. So, since it has two different alleles, it is heterozygous. We have phenotype, which are the physical characteristics, and lastly we have the genotype, which are the genetical characteristics. So how can we relate all of these terms to the picture that we have right there? So phenotype, remember that will be the physical. And both of them starts with pH, so it might be a little bit simpler for you to remember. Phenotype, physical. So the physical characteristics will be specifically the color and the flowers that we have on the left. We have purple and white. But what happens with the genotype? The genotype is basically the genetical characteristics. They both start with G, so you can remember that. So genotype and genetical characteristics. So these are different because, as you can see on the right part where there is the genotype column, you can notice that, and the combination of different alleles might cause one or another color. So at first we have both capital P's, which generates purple. We also have one capital P and one lowercase p that causes purple as well. And lastly, we have two lowercase p's that causes the white one. All right. So even though we might have a certain phenotype in an organism, that doesn't mean that the organism has both dominant alleles, such as in this one, that will be the purple color. But it can mean that it can have either two dominant ones or one dominant and one recessive. That together will form the phenotype. for that specific organism. All right, we also have something very interesting, which are Punnett squares, and it is a way to recombine genes manually to analyze its possibilities to appear in the next offspring. So, Punnett squares are very simple to use. I will teach you how to use them in a couple of seconds. But basically, what you need to understand is that you are going to recombine the alleles from the father and the alleles from the mother. And based on the results that you get in the Punnett squares, you are going to define what is the possibility of getting a certain genotype and phenotype at the end. So we're going to have capital B for brown eyes. That is one of the characteristics we're going to analyze. And we're going to have lowercase g to represent green eyes. This is going to be a real example between my mother and my father. Okay, so my father has a brown eyes and a green eyes, the alleles. That's why he has a capital B and a lowercase g. But in my mother's case, she has both a green eyes, which will be a G and G. So what happens whenever we recombine these genes? Let's analyze the possibilities. Let's start with the genes from my mother. We have G and G. And in my father's case, we have G and capital B. Okay, so what happens when we recombine the first square in there? We're going to have a capital B and a lowercase b. On the second case, we have two GGs. Then in the third one, we have capital B and lowercase g. And in the last one, we have G and G. Okay, so what does each one of the squares that we have here represent? So basically, they represent like a different child that they had. Okay, so in total, in this chart, we will be taking into consideration that they would have had 4 children. Okay, so we have to analyze the possibilities that the children from my parents will have brown eyes. And remember that brown eyes are a dominant gene over the green eyes. And we have 2 squares with the capital B's. So it means that it's possible that 2 out of the 4 children that they have are going to have brown eyes. So, which means that it will be a 50% of probability. But now, how about the green eyes? Just like you can see in the combinations right there are 2 squares that have totally recessive genes. That will be GG for both of the genes that are going to recombine for the eyes colors. So in this case, it's possible that 2 of their children are going to have green eyes out of the 4. So it means that they have the possibility of 50%. So 50% for brown eyes and 50% for green eyes. Keep in mind one more time that this is just one small Punnett square. Then I'm going to show you another example that shows us a little bit more examples on how to recombine them with more characteristics. Alright, so it's exercise time. Please complete the monohybrid practice in page number 3 of topic number 3.04. Once you are done, come back to the presentation and we're going to keep up with some explanation. Alright, so welcome back. So we're going to talk about independent assortment. And first of all, we have to analyze what the hybrid cross is. Basically, that is tracking down the recombination for 2 different traits. So remember that a trait is like the final recombination of alleles. So just like in the example that I gave you before, one trait will be, for example, a capital B and lowercase g. like brown eyes and green eyes genes, recombining both to get certain characteristics. But when we analyze the combination of both traits, which in total will lead to 4 alleles, then we have way more things to analyze. Okay. Also, we have independent assortment, which is when genes from different traits segregate independently during the formation of gametes. Remember that we already studied the meiosis process. So during the meiosis process, what happens is that the traits are going to segregate independently. All right, so now we're going to analyze the hybrid formation. So just like you can see right here, we have more genes that we need to analyze. We have dominant round, C shape. We have lowercase r, which will be recessive, wrinkled seed shape. We have capital Y, which will state for dominant yellow seed color. And lastly, we have lowercase y, which is recessive green seed color. Okay, so what's going on in here? The title says it has a RRYY and RRYY. So basically, right now we are going to track down already a specific trait with more alleles in it. So the first example I gave you about tracking the genes in the Punnett squares was very simple because we only had two alleles, but now we have way more. So let's see what we are going to do. So first of all, we need to recombine the alleles to see how many possible combinations they will get. So we have RY, we have, of course, all of them are RYs, but the difference is that sometimes they have capital letters, sometimes they are lowercase letters, which actually represent how dominant or recessive a gene will be. Once we have these characteristics, we're going to add them up in a chart to see what will happen. And right after, when we already have the 4 possibilities in the columns at the top and on the lines on the left, we're going to start recombining them and checking out what the results will be. So here you go. You have a specific recombination pattern in each one of them. So notice in the first one right here in capital RY and capital RY, when they both recombine, of course, we're going to get 2 capital R's and 2 capital Y's, okay? And at the end, this will be the resulting trait. But now what happens if we recombine, let's say, lowercase ry and capital RY? So the result will be 1 capital R and 1 lowercase r, as well as 1 capital Y and 1 lowercase y. You need to remember that each one of the results that we have right here means that they are like one baby or one offspring from the parents with different characteristics. So now we're going to do some statistics and probability analysis over the results that we have gotten in here, so we can have a more scientific perspective on what happened at the end. So we have here determining the results. Identify the genotype and phenotype of each possible offspring represented within the Punnett square. Then calculate the total percentage for each genotype and phenotype combination. So it's very important that you remember the concepts of genotype and phenotype. Remember, genotype is the genetical characteristics, and the phenotype is the physical characteristics. Okay, so let's analyze the genotype right over here. And just like you can notice right there, they have a lot of different combinations. So all of the combinations that we have in number 1, right here, like 2 capital R's and 2 capital Y's, 2 capital R's, 1 capital Y, and 1 lowercase y. All of these ones, of course, after making an average, they will get us a 6.25% of being present in the offspring. We can continue, and for example, we can analyze 2 lowercase r's and 2 capital wise. They have the possibilities of appearing of a 12.5%. And right here, they did already all of the statistics process for you. But basically, they are making an average out of the total offspring that we analyzed in the Punnett squares right there. But remember, that is merely the genetical characteristics. But how about the physical characteristics that the plants we were analyzing will present? Well, you have here that it will be noun round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled green. They will get a percent of 56.25% of appearing in the offspring. Now, let's look for a little one that's a little bit more uncommon. Wrinkled green. They will have only a possibility of 6.25% of appearing. Alright, so that's basically some analysis on what will happen in the Punnett squares. Alright, so now let's jump to your evaluation for today. Please read and summarize topic 3.04, especially focus on pages 5 to 7. Students have to create one Word document that contains all the summaries that they worked on during the unit and upload it to Google Classroom. So, I'm going to check only one Word document on Google Classroom. So, I need that you open a new Word document, and there you copy and paste all of the previous summaries that you did during the unit. They must be in order, of course. The first one, it should be the one from week number 11. Then the second summary should be the one from week number 12, so on and so forth. After you finish that, then you have uploaded it to Google Classroom as well, you have to take the quiz for topic 3.04. If you are doubting a little bit about the order in which you have to present them, you can always go back to the posts in the Google Classroom and of course notice which one was the first post, second post, third post, until you reach the last one, and you can have the correct compilation of all of them. What is the rubric I'm going to use to evaluate your summaries? I'm going to be evaluating your order, 1 point, complete content, 1 point, general appearance, 1 point, grammar, 1 point, and lastly, images, 1 point. And that is it for today's class, guys. I will be seeing you next week. Goodbye.